Okay, so first we are going to finish the answering midterm exam questions, and then I'm going to start the new topic, which is construction of phylogenetic profiles. And I'm going to skip, I'm not going to talk about neighbor joining algorithm. From phylogenetic phylogenetic tree construction, I'm going to only talk about the UPGMA algorithm because we are running short on time, so we are behind schedule. So I'm not going to talk about neighbor joining algorithm, and you will not be responsible from that in the final exam. Also I have an announcement. Maybe you may have noticed the deadline is postponed 3 days. Okay, so you are going to have 3 days to work on, 3 more days to work on the hidden Markov model construction. Now, so yesterday we started filling in the table, and we have filled in a couple of 2 of the 8 cells. Now I'm going to finish it today, and we are going to see how we can do traceback. Which are the actual sequence of states that have emitted this walk shop clean walk symbols. So the table we are filling is this. We have our states: start state, rainy state, sunny state. And we have the empty symbol and walk, shop, clean, walk. So this was the starting probability. It was 1.0 at start state, and these entries were 0 because start state do not emit anything. So it's impossible, and there's no incoming arc to start state. So there's no way to visit start state again after you have emitted a symbol. Okay, that's the reason these are 0. There is no way you are going to visit the start state again after you have emitted these symbols. After start state can only be visited once in the beginning, and then you are not going to visit it again. It's going to be between these 2 states. And since rainy and sunny states emit a symbol, it's not possible to be in the rainy state without having emitted any symbol. So these were also 0.0. And we have filled in these 2 entries, which were 0.06 and 0.24, right? Now, I'm going to fill in the rest 6 cells, and we are going to see, we are going to trace it back to find out which states were actually visited to emit these symbols. Now, to find this, so I'm going to fill in I can either fill in this one or this one, but I should go column by column, because in order to fill in this column of C, I need to know the values here. Okay, so if you write the recurrence relations, it's like this. The probability, the partial probability to be at state R and having emitted symbol S, okay, this second symbol, I need to find out, it's given by Viterbi equation, it's given me this, the emission probability of this S at this state R, and the maximum of which states can precede state R. It can be start, rainy, or sunny. So we start W, transition from start to R, transition probability. V, sunny W, and transition from sunny to rainy, or V, R, W, and transition from rainy to rainy. This is going to be V start W 0, because we cannot be at the start state and have outputted symbol W. So actually, in all the recurrence relations that we are going to fill in the rest of the table, this part is going to be 0. because it's not possible. Actually, there are some incoming arcs from the start state to the rainy and sunny states, but these arcs can only be used. These arcs can only be used when we are at start state at this empty symbol. We cannot, because there are no going back to the start state, so we will never visit start state again. So actually, when filling in these parts of the table, we can ignore, and we can just drop this out, because this one, I mean these entries, we can drop it out because these entries here are all 0. Okay, it's because of these zeros. We are sure that this is going to be 0 when filling in the, because when we are filling in this one, we are going to look at the previous column, it's 0. When we are filling in this one, the previous column here, it's 0. So it's all going to be 0. We are just going to look at these two things and choose the maximum of those. And all the things actually that we need to find this VRS is given, is known. Okay? So VSW, we know that it's 0.24. Transition from sunny to rainy, what is it? It's 0.4. So here, this one is, the top one is 0.24 times 0.4. And the other one is VRW, it's 0.06. 0.06 times transition from rainy to rainy, it's 0.7. So we are going to compare these two entities. So what is this one? 0.096. Okay, this is 0.096. And what is this? 0.042. Okay, these are like this. And 0.096 is greater than 0.042. So we are going to choose this one as the maximum. So from this, we came from this way to this cell. Okay, we can also, yes? Oh, yeah, you're right. I forgot to, so we have chosen the maximum like this, but I forgot to multiply this number, 0.096, with the emission probability. We need to also multiply with the emission probability of S shop at rainy, which is 0.4. So we are going to multiply this with 0.4 again. Okay, so it's going to be 96. This one times 4. Okay, so which is going to be which is going to be 3.84 times 10 to the minus 2. Okay. Yeah. So I can just write it like this. 3.84 10 to the minus 2. Okay. So I'm going to use scientific notation and round the multiplications to make my multiplications easier. So this is how I found the value in that cell. Now I'm going to, similarly, I'm going to, now what is the probability to be in S? I'm just going to change these to S. Now it's going to be, again, VRW. Now this time transition from R to S, and VSW transition from S to S. Okay? The only thing that is different from the previous max symbols are these transition symbols. We are still going to use the same partial probability, 0.06 and 0.24. So this is going to be, the first one is going to be 0.06 times R to S is 0.3. So it's going to be 0.0118. Okay. And the other one is going to be 0.24 times transition from S to S, it's 0.6. So it is 0.144, right? Yeah. And here again, the maximum is the one that is coming from this cell. So when filling in the second cell here, we are still going to be used the 0.241 because this 0.144 is greater than 0.018. Okay, we computed these two tables. So can you follow, or am I going fast? Well, you can follow, right? Okay. So 0.144, again, we need to multiply this one. After we have chosen it as the maximum, we need to multiply with the emission probability of S at sunny, which is 0.3. So I'm going to multiply this one, 144 e to the minus 3 with 3 e to the minus 1. It's going to be 4.32 10 to the minus 2. 4.32 10 to the minus 2. Okay? So we have filled in these two entries. We are going to continue doing the similar thing for the rest of the entries. I'm going to now do it quickly. Okay? So I'm just going to change this to C. Now I'm at this column. These transition probabilities are the same. It's going to be S. Okay, this is the Viterbi equation. This is the Viterbi recurrence relation to fill in this table entry. Okay, VSC. It's this table entry. I'm going to compare respective transition probabilities and just compare them. Which one is the maximum? Choose the one which is maximum and multiply it with the emission probability of C at state sunny. Okay, so this VRS is 3.84 10 to the minus 2. Okay, multiplied by R to S transition, which is 0.3. So it's going to be this one multiplied by 3. Okay, it's this 10 to the minus 5. Okay, I'm just going to do it like this. 1.15 10 to the minus 2. Okay, so the first is this one is 115 10 to the minus 2. The second one, 432 10 to the minus 2 times S to S transition 0.6. I'm going to multiply this with 6. It's going to be 2, 9. It's going to be this. So it's going to be 2.59 10 to the minus 2. Okay, and the maximum of these two things, I'm going to compare these two. This one is greater, so it's going to be 259 10 to the minus 2, multiply with this emission probability of clean at sunny, so it's 0.1. So it's just going to be, if you multiply the 0.1, it's going to be 10 to the minus 3. So here, this entry is going to be 259 10 to the minus 3. Now I'm going to fill in this one. VRS is equal to 384 10 to the minus 2. Transition from rainy to rainy is 0.7. I'm going to multiply with 0.7. So it's going to be 2.69 e to the minus 3. 2.69 e to the minus 3. Sorry, it's still e to the minus 2. Okay, this multiplication. It was, I multiply 384 with 0.7 for this one. I got 384 is dropped to 269. And I'm going to now VSS, which is equal to 432 10 to the minus 2. Multiply transition from S to R. Transition from S to R is 0.4. I'm going to multiply this with 0.4. It's going to be, no, I did it, so yeah, it's going to be like this. Yeah. Right, so it's going to be 173 e to the minus 2. So we are going to compare these two, 269 and 173. This one is larger. This one was corresponding to this entry, right? 384. So yeah, 384 multiplied by rainy to rainy, which is 0.7. So here to fill in this entry, we came actually from this row, okay? This one, 269 is larger than 173. 269 was from rainy to rainy transition. Okay, so we came from that cell, and 173, we are going to multiply it with the emission probability of clean at rainy. Clean at rainy is 0.5. I'm just going to divide this by 2 to multiply 0.5. It's going to be 0.86, or no, it's going to be, yeah, 0.87, okay? If you do it 0.87, it's going to be e to the minus 2. If you do it 8.7, then it's going to be minus 3. This one? No. This was 3.84 divided by 0, multiplied by 0.7. So let me actually check that again. So it was, oh yeah, you're right. This one, so I was confused. So this one, so let's do it again. So 269, actually we were going to multiply this one with 0. We multiplied the smaller number with that. So it was this was the larger number, 269, after multiplying 384 with 0.7. This is what we got. We are going to multiply this one with 0.5. I multiplied the wrong thing with 0.5. So it's going to be 1.35 e to the, so it's going to be 1.35 e to the minus, it's going to be minus 3, yes. So, and the last entries, we are going to continue. So here, this was how I'm also plotting my trace back here on the table. Now the last entry is going to be W, the last W. And this is, these are going to be, we are going to look at the previous column and R to the R, S to the R. Now, VRC is equal to 135 10 to the minus 3. We are going to multiply it with 0.7. Okay, we are going to multiply this thing with 0.7. That's R to R transition. If you multiply it with 0.7, it's going to be 0.94 e to the minus 3. Okay, or you can have it 9.45 e to the minus 4. Okay, so the first one here is 9.45 e to the minus 4. And VSC, VSC is 259 e to the minus 3. We are going to multiply with the transition from S to R. S to R transition is 0.4, so we are going to multiply this with 0.4. It's going to be 1.04 e to the 3. 1.04 e to the minus 3. Here, if we compare these two, this one is larger. Okay, this is like 10.4 e to the minus 4. So this is 10, this is 945. This is larger, so we are choosing this one. We are choosing this one means that we came to this cell from here, okay, and the final value is this 1.04 e to the minus 3 times the emission probability of W at a rainy state, which is 0.1. So it's going to be 1.04 e to the minus 4. Okay, this 1 to the 0.4 e to the minus 3 is going to be 1. Yes?
I think the VR column should be 1.35 times 10 to the minus 2.
This one?
No, the previous one.
This one. 135 e to the minus. So we are.
You multiply 3.84 times 0.7 times 0.5.
Oh, okay, you're right. It's 0.7. 0. It's going to be a 0. Yeah, you're right. This is going to be 0.2. It's not. Yeah, this should be 0.2. So that. So when we are doing this, we were. So I should consider that. I mean, I need to. After doing this correction, I need to recheck these. Which one is greater? VRC 130. So actually, this is going to be minus 3 then, right? Because this was VRC. This is going to be 10 times larger than that. So it's going to be 945 e to the minus 3. So this is larger. So I'm not actually. I will be coming from this cell here, like this. And the value that I'm going to put here is going to be just 0.1 times this. It's going to be 945 e to the minus 4. Okay, so here the final probability is 945 e to the minus 4. Now we need to also fill in this last table entry and compare these two probabilities. And whichever probability is higher, this is going to be our best path that produced the highest probability, and it's going to give me the trace back. Now, to fill in this last cell, we are just going to change these to like this. And use the same previous column values. So 135, this is 135 e to the minus 2 times RS transition. RS transition is 0.3. 0.3. I'm going to multiply this with 0.3. It's going to be 15, 0. So it's going to be 405 e to the minus 3, right? So it was e to the minus 2. Yeah, it's going to be 405 e to the minus 3. So the first one here is 405 e to the minus 3. And now let's compute the second one, 259 e to the minus 3. Actually, you don't need to do the multiplication here right now, because before even making it smaller, just 2 to the 59 e to the minus 3 is smaller than this value. Okay? So you don't need to multiply with the transition probability. It's going to, for sure, this is going to be larger than whatever you multiply this with. Okay? Because you are multiplying with a number that is smaller than 1. This is going to get smaller and smaller. So even without multiplying it with this one is larger. So it's going to be 405 e to the minus 3. And the emission probability of W on a sunny day is 0.6. So we are going to multiply it with this with 0.6. If you multiply it with 0.6, 405 it's going to be 2.43 e to the minus 3. Okay, and coming from this one, the one we computed at the top. Now, if we compare these two probabilities, we see that 2.42 e to the minus 3 is a higher probability. So that's the probability of the best path, 2 to the minus 243, 10 to the minus 3. And the states visited are sunny, rainy, rainy, and sunny. Okay? So sunny, rainy, rainy, sunny are the sequence of states that have produced walk, shop, clean, walk. Oh yeah, no. Actually, we came from this path. Okay. This one. This was 135. So this is 135, 10 to the minus 2. So this probability was generated from this probability, which is rainy state. Okay? And this probability was generated by using this probability, which is also a rainy state. And this probability was generated using this probability, which is a sunny state. Okay? So you shouldn't select the maximum at each column. You should follow the path. Okay? Because this is how you fill in this is how you do the trace back. So sunny, rainy, rainy, sunny is the sequence of states that are most likely to generate walk, shop, clean, walk actions. And you can actually verify whether your probability is true, okay? By just doing this check like this. So sunny, rainy, rainy, sunny. And I had walk, shop, clean, walk. What is walking in a sunny day? It's 0.6. So 0.6, 0.6. Rainy, rainy. Shopping and cleaning on rainy days, 0.4 and 0.5. Okay, these are my emission probabilities at these states. Also, I need to consider the transition probabilities to verify the final probability. I'm going to multiply this with the transition probabilities. My transition probabilities was So this is 0.4 from start to sunny, 0.4 times sunny to rainy, 0.4, rainy to rainy, 0.7, and rainy to sunny, 0.3. So if you multiply all these 8 numbers with each other, you should get that 2 to the 43 e to the minus 3. If you do not get that same value, it means that we did something wrong in computations. So let's actually check that. So if I multiply these two, it's going to be 0.3. If I multiply these two, it's going to be 0.24. Okay, 0.21, 0.16. Now, what is this? This is yeah, it's going to be 0.72. No, 0.072. Okay. Yeah. So this is, the emission probabilities were 0.072. And this is 20, let's multiply this with 21, 6, 1. So it's going to be 0.0336, right? If you multiply these together, this is what you are going to get. So if I multiply this with 0.072 with 0.0336, I should get something close to this number. So let's do that. Two, 9, 1, 2, 2. And it's going to be oh yeah, you're right. So actually that number is going to be close. It's actually the probability that we have computed. Okay. So we didn't do anything wrong in computations. So it's this one times 10 to the minus 4. So this is e minus 4. This is e minus 3. So the final value is this one, e to the minus 7. So it's going to be 2 to the 4, e to the minus 3. Okay? So we got the same result. So the probability is correct, and which is smaller than this, which is larger than this value. And these are the sequence of states that emitted this sequence. Okay? Any questions about this? So you are going to be performing something similar in the second, in your third assignment. You are going to be filling a table like this. But this time you're not going to be choosing the maximums. In forward algorithm, we are just going to sum up whatever probabilities we have there. We are going to sum them up to find and multiply the summation with this emission probability at the end to find the final total probability of having observed this Volk-Shop clean walk from this hidden Markov model. What we did in this question was, this Volk-Shop clean walk, we computed the probability of observing these actions through these particular sequence of states. Okay, sunny, rainy, rainy, sunny had this 2.43 e to the minus 3 probability. 10 to the minus 3 probability. So this is the probability of a specific sequence of states. If I ask the question, what is the probability of observing these actions from this hidden Markov model, you should find all possible paths, all paths that can produce these actions. You should find all of them, compute all the probabilities, and sum all these probabilities up. Okay, that's going to be the overall probability of observing these actions from this hidden Markov model. And this, again, the dynamic programming approach is going to provide us an easy way of doing this summation. You don't need to actually find all the paths, because all the paths are going to be a very large number of paths that can produce these actions. But while we're filling in this table, instead of taking the max, if we do the summation of these probabilities, it will basically mean that we are summing over all paths. Okay, so this is the only difference. Instead of using max, using a summation is going to take care of this for you in the forward algorithm. Okay, if you do not have any questions, I'm going to now start with the new topic, which is the phylogenetic trees. Let me just get the lecture slides from our course website. Okay, now we are going to start talking about phylogenetic trees. Phylogenetic trees, the main purpose, still they are related with sequences, but they can, their main purpose, they can also be generated for structures as well. You can generate phylogenetic trees for structures. Phylogenetic trees can be considered as clustering or classification mechanism, which tries to reveal relationships between a number of sequences. So we were performing multiple sequence alignment. Actually, phylogenetic trees was used as a guide tree. Which sequences are, if you remember the cluster W algorithm, first it generates a phylogenetic tree, and then it uses this phylogenetic tree to build up multiple sequence alignment by using pairwise alignments of closer sequences first, and then building, combining these sequences, multiple pairwise sequence alignments one after another. So here the guide tree was showing us which sequences are more close to each other, which sequences are far away. So given if we have a set of sequences, if a phylogenetic tree is like their family tree, which sequences are most close to each other and which sequences are far away in this family. So it's like a family tree, but it reveals, as the name, the phylogeny is coined in 1866 by Haeckel. It shows the line of descent or evolutionary development of any plant or animal species. So it's, in other words, what are the evolutionary paths, which are the hypothetical ancestors of these two organisms, whether they evolved from the same common ancestor, which two species are more likely to be evolved from a common ancestor, and these things are shown in a tree. That's hence the name phylogenetic tree. And you can also consider phylogenetic tree as a hierarchical cluster. Okay, it groups items together, closer items together first, and it continues these groupings until we only have a single group. Every organism in our set is going to have one hypothetical ancestor, and all these sets of sequences, all the species we have in this tree are evolved from this one hypothetical common ancestor. And the tree, the branches in the tree shows us the relationships of these species or sequences. There are various phylogeny or phylogenetic studies are a large part of bioinformatics studies. There are, for example, studies in the molecular biology and genetics department, for example, their students are doing their master's PhD thesis on evolutionary studies. For example, I know a couple of students who are analyzing the sheep in Anatolia, their relationship, because the sheep breed, it's really important whether they are their migration patterns, which sheep breeds came from where, whether they came from Europe or Asia. So to understand these evolutionary history, understanding evolutionary history of a certain species, we can do it by phylogenetic analysis. It can also assist, I mean, we can do historical analysis like this about, for example, about a certain species. We can talk about their migration patterns, how they evolved. We can also talk, I mean, phylogenetic analysis also helps in epidemiology, meaning that the study of diseases. For example, there's a virus, and there may be an infectious disease. And this, when this disease is infecting, spreading around the world, I mean, you are going to, by taking certain virus strains from different places, you can analyze their relationship with each other, and you can trace back where this infectious disease actually started at, okay? And I'm going to show you an example, another example about a disease in a couple of slides. And it can also aid in prediction of function of novel genes, meaning that if you put your gene, if you build a phylogenetic tree of a set of genes, some of them with known functions, some of them with unknown functions, the relationship, their closeness, their relationship in this tree is going to give you clues about the unknown gene, about the new gene's function. In other words, when the sequence search, remember the blast search? The main use of blast search was if you have a new gene, you just do a blast search, you find similar sequences, and the most similar sequence to you can be a sequence with the same function. So you can use blast sequence searches as for function prediction. Imagine you have a new gene, you do a blast search, you get a lot of results, and you look at your first result and say that, okay, oh, it's a gene with known function. It's a sequence similarity to a gene with known function, so you may use it to predict the function for your new gene. However, by just looking at the most similar sequence may sometimes give you wrong predictions, okay? There are lots of papers about this. Automated function prediction by just using sequence similarity and by just using the top hit in your blast search gives you a lot of wrong prediction. What if the situation is like this? For example, you do a blast search. This is your query sequence. You do a blast search, and these are your hits. Sequences similar to this query sequence, sorted in order of similarity. And this first gene has function A. And the remaining are, for example, have function B, and this one also has function A. This one also has function A. Okay? Now, what are you going to predict for this query sequence? What is this function going to be? Is it function B or function A? If you just use sequence similarity, simpler sequence similarity, and take the top hit, you'll probably say this gene Q is predicted to have function A. However, in that prediction, you are ignoring the fact that there are also similar sequences to this gene with function B. Okay? Now, a phylogenetic tree analysis, what it would do is, it would take all these 6 hits plus the Q, the query gene, and puts their relationship in a tree. Okay? It may be something like this. This is maybe A, function A gene. This is Q here. And it may be that I can have maybe another B here. And the other remaining 2 A's are here. Okay? It may show you that actually the first A that came as most similar to this Q is a gene with function A, but it's like an outgroup. It's like an outlier in the genes that have function A. Okay, I mean the genes that have function A is far away than the genes that have function B, and this one gene with function A, meaning that this gene contains function A, but it's actually not similar to other genes with the same function. It's in sequence, it's far away with them. And if we have this picture, we immediately see that, oh, okay, the Q gene is actually more similar to genes with function B. So such kind of a phylogenetic tree analysis is going to help you to make more correct function predictions. So that's one use of phylogenetic trees. They can aid in prediction of a function of novel genes. And there are studies, there are papers that provide different heuristics, different ideas about how to use the phylogenetic tree that is built from similar sequences. It's used in other biological studies as well. There's also the idea of, in phylogenetic analysis, when we are thinking about species, there's also mitochondrial DNA material is used a lot in phylogenetic studies, because mitochondrial DNA does not go over crossover. Okay, mitochondrial DNA you only inherit it from your mother, so you can use maternal lineage can be found easily by using mitochondrial DNA. So it's mitochondrial DNA is not in nucleus, it's in mitochondria, and it's like a circular DNA. It's like a DNA of a prokaryote, okay? Small circular DNA. And it codes for 13 proteins, 22 tRNA genes, and 2 rRNA genes. And it has also another thing from nuclear DNA in compared to nuclear DNA is it has higher mutation rates than nuclear DNA. It means that we can study in we don't need to go back too much in history to find out how these species are related. I mean, so even for closely related individuals, there are going to be differences in mitochondrial DNA due to mutation rates. But there's no crossover, so it can be more. It can give more clues about their evolutionary relationships. Phylogenetic trees can be used to understand the origins of HIV virus, okay, how different strains of HIV, how they are related to each other, how the HIV virus mutated and evolved in time, and it shows the relationships. As you can see, this tree is unrooted. There is no root of this tree, meaning that there is no single node. I mean, you can take anywhere and denote it as a root. And another thing that is important here in this tree are the branch lengths. Okay, the branch length in this tree is important. It tells something. For example, why is this virus, this strain, very far away? I mean, we could have put this. I mean, if we were just talking about an abstract tree with just nodes and edges, I could also put this one in this layout. I could just put this label. I could cut this branch short, and I could put this label here. Here in this visualization, the branch lengths are taken into account, and the sequence distance, their sequence similarity, the branch length actually shows their evolutionary distance. So it shows that this strain is very far away from the other strains. These two strains are closer to each other. So a phylogenetic tree may give ideas about evolutionary distance. These distance information can be integrated in the tree. And it shows us a hierarchical relationship of these entities. It can also be used to understand relationships between species. So from the molecular view was different. I mean, this phylogenetic tree is different than the phylogenetic tree that is built by using mitochondrial DNA. If you look at the mitochondrial DNA of these species, we see that the humans are not the out group here. Humans are in a subgroup between chimpanzees and bonobos, okay? And orangutans are the different species in these groups of 5 species. And phylogenetic trees can also help in real cases. This was a criminal investigation, okay? Which tries to, so here the criminal investigation, it was there was a case about a Florida dentist, okay? The Florida dentist infected his patients with HIV. So to find that out, they had certain local control, meaning that people who have HIV, but they are not patients of this dentist, they also got blood samples from the dentist, and when they do the phylogenetic tree of the HIV strains, okay, the blood samples of these local HIV infected people and dentist patients, we see that dentist patients are grouped together here. So they are different than local control. So they got HIV, and the fact that they got HIV, they're all related, means that they probably got the HIV virus from the same source. And what ties these groups of people are, they're all patients of the same dentist. So it helped the case against this dentist. Okay, they fall within the clade of HIV sequences found in the dentist. But some patients here, they are also not belong to the same group. So maybe some of the patients of this dentist have HIV from other sources. And I'm going to skip. We already went over what orthology and parology. We went over these evolutionary events. I'm going to skip these biological preliminaries. Now I'm going to just continue with these, how to build phylogenetic trees. I'm going to talk about UPGMA algorithm after we have a 10-minute break.